Hi and welcome back to the cell. We are officially starting our quest into the cell, and we're going to start studying all the different organelles that operate within a cell. And on the website as well, I have a picture of an animal cell and another one of a plant cell. And the plant cell is more rectangular in shape. And it's just a bunch of lines pointing to them. And what I want you to do is to try and identify all these different things inside the cell. These are the organelles we are going to study. So what I would like for you to do, if this helps you as well for studying, is as we go through our lectures, I label the picture, draw descriptions of the organelles, and hopefully that'll give you a visual to where we are inside the cell, how these different organelles are relating to each other, and just give you a way to identify these different structures inside the cells. These may not be the best pictures for you. You could always go online and find some more, or in a textbook for sure, but just wanted to give you something available at your fingertips in case you needed something. All right, so let's start studying these organelles. This is where it gets really, really interesting when you start getting into the inside of a cell. So like I said, we're going to stay with eukaryotes and go into those organelles. So eukaryotic organelles. And if you want to define organelles, the best way to define an organelle, these are specialized compartments within the cell that serve a specific function. So being specialized means that they have some kind of specific function. So specialized compartments in the cell. We're going to begin with an organelle we've already talked about just a touch, and we're going to start at the center of it all, the nucleus. The nucleus. So the nucleus is, we've already defined it a little bit, it houses the genetic material for the cell, the DNA. So houses, it's kind of its job to houses the genetic material for the cell. And the nucleus is a little bit more special than other organelles, mainly because it's carrying this precious cargo. Our DNA never, ever, ever leaves the nucleus. And so the DNA has to stay inside the nucleus. It's kind of like the blueprints to a building, the original blueprints to a building. You never take the original blueprints out of the office. You make copies and send them to all the different contractors that are building this building. That's kind of how DNA operates as well. When we get to our molecular biology unit, this will start to become more and more clear. But what we need to know for the nucleus, it's a membrane-bound organelle, and I alluded to this last time, but it's a little bit more special than having just a phospholipid bilayer. It actually has a double phospholipid bilayer for a membrane, and that membrane around the nucleus is called the nuclear envelope. So the nucleus is surrounded by a special membrane called the nuclear envelope. And what's neat about the nuclear envelope, it's made of a double phospholipid bilayer. We're going to draw this out. Double phospholipid bilayer. All right, so here is my typical cell. And I'm not going to draw the other organelles yet. We'll just keep it simple right now. And here's my nucleus. All right, inside my nucleus is all the DNA in there, and I'm housing it, keeping it nice and happy. So everything's good going on in there. If I were to zoom in on this nuclear envelope right here, here's what this would look like. I'm just going to take this piece right here and kind of blow it up for you. What you'll see is a double phospholipid membrane, but you can imagine things have to go in and out of the nucleus. If I'm DNA, I need to send message out to the cell saying, do this or don't do this, and I need access and delivery points where I could send messages in and out of the nucleus. And just like our cell membrane is studded with all these proteins that allow for transport in and out, the same thing is going on with the nuclear membrane, but it has to go through 2 layers of the phospholipid bilayer. So give me patience as I draw this really as quickly as I can. Maybe you can start to see what's going on here. Okay, that's the double phospholipid bilayer. I'm hoping it comes through pretty clearly. So here is one phospholipid bilayer, here is another phospholipid bilayer. Together, you get your double phospholipid bilayer. This huge protein structure that's providing this channel for passage of specific things. It's not just anybody who wants to go back and forth. This is called a nuclear pore. And there's other types of proteins that reside on a nuclear envelope as well, but this is a specific one that forms like this barrel-like shape in the membrane. What you're seeing is a cross-section of it. So really, if you're looking at it this way, that's what's going on right there. And this nuclear pore allows for specific passage of things inside and outside of the cell. And there's obviously, you got 2 phospholipid bilayers. One of the inside phospholipid bilayer stays around the whole entire nucleus, and this one that's on the outside facing the cytoplasm side, the space inside the cell, that one is going to start interacting with other organelles, and we're going to get to that one later. But know that the nucleus has a double phospholipid bilayer. That is the only time you'll see that in the cell. This one's special. And it has to be because it's carrying our genetic material. So we need to really, really protect it. And the nucleus has to interact with other organelles, and using a membrane is actually one way to do that. So you're going to see that coming up soon. Okay, so that's the nucleus. Main job is to house the genetic material for the cell. The next organelle we're going to talk about is the ribosome. Now the ribosome is something a little special too. They're all special. Can't help it. But the ribosome is not a membrane-bound organelle. So let's slow down here for a second because I'm going ahead of myself. The ribosome. Ribosome. This is a non-membrane-bound organelle that is responsible for producing protein inside the cell. So non-membrane-bound organelle. Responsible for making protein inside the cell. All right, so here's where I want you to think a little bit deeper. If prokaryotes are allowed, basically do not have membrane-bound organelles, can they have a ribosome? Yes, because a ribosome is a non-membrane-bound organelle. We have ribosomes, they have ribosomes, they just look a little bit different. So this is present in both prokaryotes and eukaryotes. They're just really small in prokaryotes and a little bit larger in eukaryotes. This is the only organelle that you will find in both types of cells. So present in both prokaryotes and eukaryotes, responsible for making protein in the cell. But also, I want you to know that ribosomes, they're obviously different. They're not membrane bound, they exist in both, but here's why. They're made of the stuff cells are made of, and here's what they're made of. They're made of RNA and protein put together. And when you put RNA and protein together, they create this beautiful specific structure. In this case, they'll make a ribosome. You could do other structures as well, but in this case, you're making a ribosome. And this ribosome now has the catalytic activity to create proteins. And so it links all those amino acids together. And that's the protein machinery of the cell. So it's made of RNA and protein. And this provides catalytic activity. It's actually the RNA that's doing the catalytic activity in this whole entire structure, but the protein is working with the RNA so it can do its job. But you don't need to worry about that detail yet. But what I do want you to know is that it's present in both, it makes protein, and here's the specifics on how ribosomes, or the types of proteins ribosomes make. There are two types of ribosomes. You have bound ribosomes you can think of what the opposite is already. So bound ribosomes are ribosomes that are embedded in the membrane of another organelle, and this organelle is called the endoplasmic reticulum. We're going to get to that one next lecture. So bound ribosomes, or a couple lectures from now, bound ribosomes are ribosomes. These guys are little and small. I want you to think of them as like, you know, if you have a meal and you sprinkle some pepper on it, the pepper would be the ribosomes. They're really, really tiny compared to the size of your meal. So ribosomes, so for bound ribosomes, ribosomes that are embedded in the membrane of the endoplasmic reticulum. That's an organelle. We're going to get to that. So don't worry about not knowing what endoplasmic reticulum is yet, but it's a special organelle inside the cell. And remember how organelles are surrounded by membranes. The endoplasmic reticulum membrane, if there's ribosomes inside that membrane, that ribosome is now considered a bound ribosome. Bound ribosomes, they produce proteins that either function in the membrane, it could be an organelle membrane, it could be the cell membrane, or they produce proteins that get secreted out of the cell. So if I'm a cell and I need my protein out because it needs to go talk to another cell, I'm going to make that protein from a bound ribosome. So these guys produce protein that either function in the membrane, I'm going to say in a membrane, because that membrane can be anything from the nuclear envelope membrane to the cell membrane to even the endoplasmic reticulum membrane. It doesn't really matter what membrane. It will know its function when it's made, and it'll go to that specific membrane. It'll produce proteins that either function in a membrane or that get secreted out of the cell. All right, so I'm going to erase this stuff up here, and we're going to do the other type of ribosome. So if you got bound ribosomes, the opposite of bound would be free. So in biology, we like to do the opposites all the time. So if one does one, the other one does the other. That's how bio works usually. So, little b would be free ribosomes. And just like the name suggests, these ribosomes are literally free. They are hanging out in the cytosol of the cell, the nice fluid inside the cell, just hanging out in that. It's like they're just sitting in a pool for just waiting to be used and make protein. So these ribosomes that function in the cytosol. Okay, now these types of proteins that get made from the free ribosomes, these proteins, it's kind of like, I want you to think about it, where you reside is where you kind of work to a certain degree. So bound ribosomes make proteins that either work in a membrane because they were made kind of bound in a membrane, or they have to be secreted out of the cell, they have to pass through a membrane. Free ribosomes that make protein, these guys make protein somewhere in the cytosol. These proteins are also going to function in the cytosol. So it produces proteins, oops, I was going to say produces proteins that function in the cytosol. All right. So I want you to think about it. Let's pretend, I'm going to ask you, would this type of protein be made from a free ribosome or from a bound ribosome? Okay. So if I needed, let's go back to our last section. What if I needed an aquaporin to be made? Remember what an aquaporin is. It's that membrane-bound pore protein that allows water to go in and out. If I need to make an aquaporin, what type of ribosome would help make that protein? A bound ribosome or a free ribosome? A bound ribosome, because bound ribosomes make proteins that function in the membrane. Let's see. If I need to make, all right, there's certain kinds of proteins that we call them our skeletal proteins, our cytoskeletal proteins. We're going to get to those too. But these guys help create the shape of the cell, and they just kind of swim around in the cytosol. If I'm making a protein that functions in the cytosol, I need a bound or a free ribosome. A free ribosome. Let's do another one. Let's say I'm an intestinal cell, something in your gut, and food has now come into your stomach, into your intestinal tract, and you need to send out these proteins that help digest your food. So if your cell is here and the food is outside your cell, you need to send these proteins outside your cell so they can start digesting the food. You're secreting these proteins. Do you need a bound ribosome or a free ribosome to start making these proteins? A bound ribosome. So I want you to start thinking like that so you could figure out, oh, that's from a bound ribosome, that's from a free ribosome. Just so you could start understanding the beautiful nature of protein production. Now here's what's really neat. Ribosomes can switch from being bound to free and free to bound. They could just like plop themselves into the membrane of the endoplasmic reticulum and then remove themselves if they need to. And so they're very flexible when it comes to making protein. They will go wherever they need to go because they will actually kind of read the message that has been given to them from the DNA saying, I need to make more free or more cytosolic proteins that function in the cytosol. Then that ribosome is going to stay in the cytosol or put itself into the cytosol to make that protein. So it knows how to read instructions really, really well. All right, so that is it for nucleus and ribosome. Come on back, we have more organelles to go through, and we're going to get to some really fun ones too. So I will see you soon.